Hello everyone, and welcome to our second screencast on the immune system. Today in our discussion we're going to talk about the acquired defenses. Recall that we talked about at the beginning that the acquired defenses represent for us a third line of defense. Remember what the first two lines of defense were? The barriers to entry and the inflammatory response, or the inflammatory response involving the white blood cells, neutrophils, and macrophages represented that second line of defense. So we're going to talk about acquired defenses, the third line of defense here today. Unlike the first two lines of defense, which basically work just about against any antigen that enters the body or potentially wants to get in, the acquired defenses are very much specific, which means that they recognize and are directed against only a particular pathogen at any one time. So acquired defenses will work against this pathogen but not that one. Whereas the first two lines of defense work generally against all pathogens. In addition, the acquired defenses are systemic. They're not restricted to the initial site of the infection as we saw with the innate and particularly the inflammatory response. So they're going to occur system-wide or throughout the entire body. They can go anywhere the pathogen is, travel to the pathogen, and have an effect on the pathogen. In addition, acquired defenses also have memory. As we're going to see as we go along, we'll talk about how that memory develops. But that means they have the ability to, once a pathogen enters the body, to recognize and mount even stronger attacks on the pathogen if the pathogen happens to enter the body again. Okay, so there are two components to acquired defenses. These two components are called the cell-mediated response and the antibody-mediated, or as we'll see later, the humoral response. Now, we're going to talk about these two responses and these two components of the acquired defenses separately, but I want you to know that they both occur simultaneously. So once the acquired defenses are engaged, both the cell-mediated and the antibody-mediated immune response will be occurring at the same time. Although we're going to separate them out and talk about each one separately, just keep that in mind. Okay, so we'll talk about cell-mediated immunity first. Before we actually get into the nuts and bolts of cell-mediated immunity, though, there are a couple of things that I just want to make sure we give you some background on it that you understand. The first is macrophages. Now, we've already talked about macrophages when we talked about the inflammatory response. Remember that macrophages are large amoeboid cells. That means they can move around, they can crawl around through the body tissues to find pathogens, engulf and remove them. And they do exactly that. Once they encounter the pathogen, they can phagocytize, engulf, or remove the pathogen. Now, an important term we have to take a look at that's associated with these macrophages, and to tell you the truth, with all cells in your body, are what we refer to as MHCs, or major histocompatibility complexes. These major histocompatibility complexes are proteins that are located on the surface of all of our body cells, and they are unique to each individual. No two individuals, possibly with the exception of identical twins, have the exact same major histocompatibility complexes on the surface of their cells. So everyone who's watching this video, no two of us, including myself, have exactly the same major histocompatibility complexes that stick up off the top of off the surface of their cells. They extend up from the membrane of the cell. And what they really do is tag that cell as belonging to the body, so that the immune system knows as it surveils the body which cells belong there and which cells or potential pathogens do not belong there and can distinguish between them by looking for the major histocompatibility complexes. So again, these major histocompatibility complexes with regard to the macrophages, just like all cells in the body, are going to identify macrophages as being normal body cells that belong there, and no immune response would be initiated against them. Now, when a macrophage, however, phagocytizes a microbe, it will take fragments of that microbe, send them to the surface of the cell, attach them to what are called class 2 major histocompatibility complexes, and present them on the surface of that macrophage. This is going to now say that there's something going on with this macrophage. It's not like it was before, and it's actually now presenting an antigen to the immune system, and as a result of that, an immune response is going to occur. In cell-mediated immunity, this is going to occur when T cells recognize the macrophage now as an intruder because of the class 2 major histocompatibility complexes and the fragments of the pathogen as an infected cell. And that immune response is then going to take place. So let's take a look at how this works. So here's a macrophage. This is a typical macrophage in the body. So this is a normal body cell. And as such, it's going to have these proteins on its surface which tag it and identify it as being a normal body cell. These proteins are called major histocompatibility complexes. And since they're present on the surface of the cell, they're referred to as class 1 major histocompatibility complexes. Now, in addition, macrophages, because they're part of the immune system, also have another type of major histocompatibility complex. These are called class 2 major histocompatibility complexes, MHC2. Now, these aren't present on the surface of the cell. They're found in the cytoplasm of the cell. And they're the ones that are going to be important to setting off the immune response. So let's draw another cell here, another macrophage. But this time, with this macrophage, we're going to ignore the class 1 major histocompatibility complexes because we know they're there. But to save and maybe not confuse you, we're going to eliminate them and just concern ourselves with the class 2 major histocompatibility complexes. So now let's say that a pathogen enters the tissue of the body. So here's our pathogen. What's the job of this macrophage? It is going to recognize the pathogen as being foreign. It is going to move to the pathogen, and macrophages are phagocytic cells, so it's going to move over, phagocytize, and engulf the pathogen. Now, let's make another drawing over here then. Here's our macrophage now with the pathogen engulfed by the cell. So the pathogen is now going to be located in a vacuole. Here are our class 2 major histocompatibility complexes. And now what's going to happen is lysosomes will fuse with this vacuole, secrete enzymes in that will digest and destroy the pathogen. So the pathogen will be broken up into fragments. Once the pathogen is broken into fragments, fragments then will be transported from the vacuole to, and they will attach with the class 2 major histocompatibility complexes. once the pathogen binds to the major histocompatibility complex. The class 2 major histocompatibility complex will be transported to the surface of the cell along with the pieces of antigens that have been attached to them. And that macrophage will now present the antigen on its surface. This macrophage is now referred to as an antigen presenting cell. It's been infected, essentially, by the pathogen and now is presenting pieces of the pathogen on its surface. This cell, this antigen-presenting cell, is going to be critical to setting off the remainder of the immune response. Okay, so now let's take a look at the kind of nuts and bolts of cell-mediated immunity. As the name suggests, this is going to involve cells that are going to be engaged in the immune response. We have several types of cells that are engaged. The first type we'll take a look at are what we call helper T cells. Helper T cells will combine with macrophages in the lymph nodes. So when a macrophage picks up, engulfs, and destroys a pathogen, it presents fragments of the pathogen on its surface by the major histocompatibility class 2 complexes. And now that, as we saw in the video, now that macrophage is going to enter the lymphatic vessels and travel to the lymph nodes. In the lymph nodes reside the lymphocytes, right? Two types of lymphocytes, B cells and T cells. We're going to be focusing here in cell-mediated immunity on the T cells. Here's our first type, the helper T cells. So when that macrophage enters the lymph node, it's going to combine with a T cell. It's going to kind of dock with it. And once it docks with the T cell, the helper T cells will then be stimulated to produce a set of highly reacted proteins, and these proteins are called cytokines. So these T cells exist in the lymph nodes, these helper T cells exist in the lymph nodes, but they're just kind of sitting there dormant, if you will, not really doing anything until they encounter antigen-presenting cells. Once that encounter takes place, it sort of engages the helper T cells, they become activated, and they begin to produce these highly reactive proteins called cytokines. These cytokines then, once produced by the helper T cells, will then stimulate various other T cells into action. So that's why they're called helper T cells, because they help to stimulate the remainder of the immune response. Now, in addition to helper T cells, there are also what are called cytotoxic T cells. These cytotoxic T cells can be activated by these cytokines. As a matter of fact, what happens is the helper T cells actually kind of differentiate into cytotoxic T cells. Now, these cytotoxic T cells basically have two functions. One function is to kill macrophages that are being displayed by the antigen that are antigen-presenting cells, I guess I should say. So the antigen-presenting cell has come in, it's engaged the helper T cell is activated, helper T cell produces the cytokines, differentiates into a cytotoxic T cell, and the cytotoxic T cell now will kill the macrophage that's displaying the antigens or the antigen-presenting cell. In addition, the cytotoxic T cells can secrete additional cytokines of their own, and these additional cytokines can stimulate additional macrophages, sort of kind of saying, hey, there's antigen out there, there's pathogens out there causing damage to the body, and the cytotoxic T cell now recruits more macrophages and says, get out there and engulf and remove those pathogens. So let's take a diagrammatic look at cell-mediated immunity and what we were talking about textually there. So here, this is going to represent our APC, our antigen-presenting cell. So the antigen-presenting cell is going to move back into the lymph system and travel to the lymph nodes. Once the APC is in the lymph nodes, let's draw it in the lymph nodes now, the APC will encounter the lymphocytes. Here's a T lymphocyte. This is going to be a T4 or a T helper cell. Now these T4 or T helper cells themselves have, let's call them proteins that kind of act as docking mechanisms. And what's going to happen is this antigen-presenting cell is going to be able to dock with this T4 cell or this T helper cell. Once it docks with the T helper cell now is going to be stimulated, it's going to be activated or stimulated, and it's going to begin to release highly reactive proteins called cytokines. Once these cytokines are released, the cytokines will seek out and bind to the surface of macrophages present in the lymph node. And these cytokines, once they bind, will activate these macrophages. Once activated, the macrophages will then move into the bloodstream, travel to the site of the infection, and continue to engulf and remove the pathogens from the tissues. In addition, once the cytokines are released, this T4 cell now will also begin to differentiate. These T cells will differentiate, and they will differentiate into another type of lymphocyte called a cytotoxic T cell. So there we have our cytotoxic T cell. Now, once the cytotoxic T cell forms, it'll do one of two things. It will either recruit additional macrophages, which will be stimulated by the cytokines, or another job of this cytotoxic T cell is to induce apoptosis of the APCs that are coming in. So what it'll do is the cytotoxic T cell will go to the original antigen presenting cell and say, kind of, thank you very much. We appreciate what you've done for us. Now you need to be destroyed. You've completed your job. Okay, now that we've taken a look at the video of how the helper T cells engage the antigen presenting cells and how the cytotoxic T cells work, it is important for us to note that there are other types of cells, T cells, that are involved in the cell-mediated immune response. One of these types are what we call natural killer T cells or killer T cells. These have a very similar function to cytotoxic T cells. They react basically the same way. However, they're the primary mechanism of defense against tumor cells, against cancer. And then finally, we have what we call suppressor T cells. Suppressor T cells, their job is to dampen the activity of the cytotoxic T cells and the NKT or natural killer cells as the antigen stimulus lessens. So you can imagine that what's going to happen is as the antigen presenting cells come back, engage the helper T cells differentiate into cytotoxic T cells. They recruit more macrophages. The macrophages are going to go out, help the neutrophils, and eventually the pathogen is going to be removed from the system, and the amount of pathogen is going to go down, down, down. So as it goes down, there are going to be less and less antigen-presenting cells coming back to the lymph nodes. So suppressor T cells then recognize that this is happening, and they then will secrete proteins, cytokines themselves, that will then tell the cytotoxic T cells, okay, you've done your job, the pathogen's been cleared from the system, you don't have to recruit macrophages anymore, it's time to just calm down and lessen your activity or suppress the activity. Okay, the second component of the acquired immune defenses is antibody-mediated immunity. And I want to emphasize again here that as we go through this, you should recognize that both divisions of the acquired immune defenses are happening simultaneously. This is kind of all happening at the same time, even though we're talking about it separately. So in antibody-mediated immunity, this is also known as humoral immunity. And the reason why it's called humoral is not because this is the type that's kind of funny to watch, but because it occurs in the blood, one of the body humors, okay, along with the lymph and other body fluids. So body fluids are referred to as humors, and the blood is one of them. So antibodies can be found in the blood. This is also referred to as humoral immunity. Antibody-mediated immunity, unlike cellular immunity, though, is going to primarily involve the B cells. And the B cells are going to produce these antibodies in response to foreign antigens. The mechanism of action, that is to say, how the B cells work, is oftentimes described as the clonal selection model. You'll see why in just a minute. So I'm going to break it out step by step, but remember, this is all happening kind of in rapid succession. So to understand how antibody-mediated immunity works, first we have to have an antigen. So an antigen is going to enter the lymphatic organs or the lymphoid organs, maybe the lymph nodes, let's say, for example. In the second step, once the antigen enters, it is going to bind to a specific B cell. We'll show you a video in a minute of how this works. And that B cell has to have the correct antibodies on its surface. If it has the correct antibodies on its surface, the antigen will be able to bind to it. Once the antigen binds to the B cell, it sort of activates the B cell, kind of turns it on. The B cells, like the helper T cells in the lymph nodes, are just kind of sitting there, again, dormant, if you will, until the antigen shows up. Once the antigen shows up and binds, that signals the B cell, you got to kick into action. So once the B cell becomes activated, it then will combine with a helper T cell, and the helper T cells will then begin to secrete the cytokines. Now, if you look here on the PowerPoint, I have the term lymphokines. Remember, the T cells are lymphocytes. So these highly reactive proteins that they produce, we could call them, because they're cells, cytokines, or another term for them, I'm just introducing it to you here. I really don't want to confuse you, but in the literature you may read the term cytokine or lymphokine. So I want you to know that they're really the same thing. Lymphokines then are just cytokines that are produced by the helper T cells. Once the T cells secrete the lymphokines, the activated B cell itself, let's turn our attention back to the B cell now that docked with the T cell. The B cell, in response to the cytokines, is going to begin to undergo mitotic divisions, and these cells will differentiate. So what will happen is The B cell will bind to the T cell, helper T cell. Helper T cell will release cytokines. The cytokines will stimulate the B cell to undergo rapid mitotic division. By the way, that would be called clonal expansion because each of the daughter cells are really clones of that original B cell that docked to the T cell. And again, you'll see that in the video. Once these plasma cells form, or once these cells divide very rapidly, they will differentiate into one of two types of cells: plasma cells or memory cells. Plasma cells will go on to produce antibody, while memory cells will remain in the system, and they will have the ability to produce antibody, but they don't produce it immediately unless there is a second infection with the same pathogen. So let's take a look at a video on how this works. Okay, so let's take a look at a visual representation of antibody-mediated immunity. You know, in the first step, we said that antigens enter the lymphoid organs. Well, the question becomes, where are these antigens come from? Well, I want you to think back or recall for a minute whenever we talked about pathogens invading tissues. Once they got into the tissues, what was the initial response? Initially, two cells enter the tissues and attack the pathogens. One of these cells, we said, were the neutrophils. As a matter of fact, we said they were the first ones in, and they encounter the pathogen. They will engulf or phagocytize the pathogen, and then once the pathogen is inside, they'll digest the pathogen. And destroy it, digest it down into little pieces. But these neutrophils are pretty small cells, and the pathogen's almost the size of the cell. As a result, the neutrophils, we said, they die. And whenever they die, they then will release fragments of the pathogen. And those fragments of the pathogen will then enter the lymph vessels, and then through the lymph vessels will eventually be passed to the lymph nodes. So in the lymph nodes, we have the appearance of the fragments of the pathogen. These fragments are the antigens that we're talking about that enter the lymph nodes. Now, remember, just for the sake of understanding and trying to put it all together, there was a second type of cell, which we already talked about, that enters. It's actually kind of called in by the neutrophils. It enters the tissues a little bit later, and that is the macrophage. So what happens? The macrophage comes in, it engulfs the pathogen just like the neutrophil did, but what's the difference? The macrophage is able to digest the pathogen into individual fragments, but the macrophage doesn't die. Instead, it takes those pieces of pathogen and presents them on the surface via major histocompatibility complex class 2, and the macrophage then becomes an antigen-presenting cell. That antigen-presenting cell then enters the lymph vessels, travels to the lymph nodes, and enters the lymph node as an antigen-presenting cell, okay, which we've already talked about. And that's going to set off the cell-mediated response. But now what we want to talk about is once these antigens enter the lymph nodes, that's our first step, what happens next? Okay, so what happens next? So let's take it from there. So here's our antigen now that has entered the lymph node. Within the lymph node is where the B cells reside. And the B cells form various colonies. So each colony of B cell expresses proteins on its surface called antibodies, and each colony expresses a different kind of antibody. So as we can see here, this top B cell is expressing an antibody that has a receptor on its surface that has this shape. This one has an antibody that has a receptor on its surface with this U shape, and this bottom one here has a antibody that's expressing a receptor that has this diamond shape. So this antigen can only bind to one of these three different types of B cells. Which one can it bind to? Obviously, it can only bind to the B cell that has the correct receptor on it. So it's going to be able to bind to this middle one, not the top one, not the bottom one. Okay, so now once that happens, let's draw it over here. So here's our correct B cell with the right antibody on its surface. Here it is. And there's the receptor. Now the antigen is going to bind to the receptor of that B cell. With that receptor binding, once the antigen binds to the receptor on the antibody, that B cell is going to be activated. And once it's activated, it is going to begin to express antibody. What antibody? Antibody with the same exact receptor on its surface that the activated B cell has. So let's draw some antibody. So this guy is going to now start to express these antibodies. So there are some of the antibodies that are expressed. What will happen to these antibodies? Those antibodies will enter the blood and travel to the infected tissues. Now, in addition, once this B cell is activated, it will also process this antigen, and then it will attach the antigen to a major histocompatibility complex again. This is going to be major histocompatibility complex 2, class 2, and it's going to then become an APC like the macrophage was. Now, a T cell, let's draw a T cell here, and its docking mechanism can come over and interact with that B cell that has now become an antigen-presenting cell. Once the T cell binds to it, what does the T cell do? It begins to express or release cytokines into the system. And these cytokines will make their way to the surface of the B cell and bind to the surface of the B cell. With that binding, the B cell will begin to undergo mitotic divisions. And the B cell, 1 will become 2, 2 will become 4, 4 will become 8, so on and so forth. So what will happen is we say that there will be a clonal expansion of that particular B cell. It'll clonally expand. These are all clones now of this original B cell that became the APC. Now, what will happen? Just like this one, these cells will now go on to produce antibody. What antibody? The same antibody that the original B cell was producing. Those antibodies will then be released into the blood and travel to the tissues. Okay. Now, with this clonal expansion, these new B cells will be able to produce antibodies at the rate of about 1,000 per second. So the amount of antibody, once this clonal expansion occurs, will go up very, very rapidly. These cells that produce the antibodies we refer to as plasma cells. Now, not all the cells that result from these mitotic divisions will become plasma cells and produce antibody. So let's draw an extra one in here, and this one doesn't actively produce antibody. So this one kind of remains in the system, and it knows how to produce the antibodies, but it isn't producing them now. It becomes what we call a memory cell. So that if you get reinfected with the same pathogen somewhere down the road, these cells have memory for that pathogen and they will immediately start producing antibody. As a result, you may not even know that you've been reinfected because your immune system very quickly with these antibodies can take care of the pathogen and eliminate it from the system before it can cause any damage. By the way, as a side note, how do these antibodies work? Well, generally, once they enter the blood and travel to the tissues, these antibodies can bind to the antigens on the surface of the pathogen, let's say, for example, a bacterium. And when they do that, they mark the bacterium for removal by the neutrophils and the macrophages. Initially, the neutrophils and macrophages are pretty slow at eliminating the pathogen from the body. But once the antibodies bind to the pathogen, that we say opsonizes or makes it much easier for the neutrophils and the macrophages to engulf and remove the pathogen from the system. So they now work very rapidly to eliminate the pathogen from the system. So the plasma cells, one thing I forgot, the plasma cells are going to secrete the antibodies. I did mention that. Okay, so we've been talking about antibodies up to this point in time. We should know a little bit more about them. Actually, we take a look at antibodies. There are 5 important classes of antibodies, and you can see them on the slide here. They are listed as IgG, IgM, IgA, IgD, and IgE. Now we take a look at them. Why the designation Ig, G, and then M, A, D, and E? That's because antibodies, you may recall we talked about this when we were going over the blood, are also referred to as immunoglobulins. That is to say, globular proteins that are involved in the immune response. So, since another name for an antibody is immunoglobulin, the different types are tagged Ig, G, M, A, D, and E. Now, we don't have time in this class to take a look at all the different types Maybe that's something, if you take immunology, or immunology is a part of microbiology for you, you'll be able to take a closer look at. But I want to focus on just two of the 5 classes of antibodies here in this class. The first one we're going to focus on is IgG. This is the most common antibody that can be found in the blood. It's composed of 2 long chains and 2 short chains of amino acids that have kind of a Y shape to them. That's why I've been drawing antibodies with that kind of Y shape that you've been seeing in the past slides and videos. IgG antibodies, one end of the antibody is said to be variable, and the other end is said to be constant. The variable side is the side that's designed to be able to bind to a specific antigen, while the control or the constant side is kind of the side that will, let's say, for example, anchor into the B cells or be free in the blood. Okay, our second class of antibody that we'll take a look at are the IgM antibodies. These antibodies are present on B cells in lymphoid tissue. So, when we were taking a look at that explanation video of the B cells, those antibodies that were sticking up off the surface of the B cells are actually IgM antibodies. IgM antibodies are the largest of all the antibodies. They're actually groups of 5 monomers linked together, 5 distinct individual antibodies that are kind of linked together. And IgM antibodies are the ones that are released into the blood first. So if IgM antibodies are the ones that are released into the blood first, why are IgG the most common? Well, here's the thing. If you recall in the video, the plasma cells, once the B cell expands, mitotically expands and divides and forms the plasma cells begin producing antibody. The initial antibody that the plasma cells produce is IgM antibody. But later, as the response continues, the plasma cells sort of switch from the production of IgM to the production of IgG antibodies. These IgG antibodies are much better at recognizing the antigen than the initial IgM antibodies are. So the IgM antibodies are released first by the plasma cells to get a kind of a quick development of antibodies into the system, but later they're going to be replace the IgM antibodies by IgG. So what's the importance of this? Well, it can help us to determine whether a person's in an acute or chronic stage of an infection. If it's an acute stage, it's recent, there's going to be a higher concentration of IgM antibody in the blood. However, if it's long-lasting or chronic or has been an infection that's occurred in the past, we're going to see a higher concentration of IgG. These IgG antibodies will remain in the system for a longer period of time as well than the IgM. The IgM, we say, have a shorter half-life than IgG antibodies. Okay, so here's that structure of the antibody that I was talking about. Has its characteristic Y shape, and we can see here's the light blue represents the constant region of the antibody, and then at the tip of each side we have the variable region of the antibody. Notice the variable region has a certain shape to it, and the shape of the variable region determines what antigen that antibody is able to bind. So for example, this antibody can bind this yellow antigen, but it can't bind purple, red, green, blue, or beige, because if you notice, they don't have the right size or the right shape to be able to fit into and therefore bind with the antibody. So different pathogens are going to have different antigens that are going to be produced, that are going to be recognized. So this antibody can recognize a pathogen that produces this antigen, but not this antigen. Okay. This is a video that you can watch. I'm not going to go over it here in the screencast, but this is a video that you can watch. Remember, we talked about natural killer T cells give us defense against cancer cells. And the question becomes, well, then why do people get cancer if we have an immune system that can destroy cancer cells? This video is going to help you to be able to understand that. So take a look at that video. It is in Canvas, and it is under the videos and animations segment. I think you'll find it very interesting. And it also talks about immunotherapy and how we're using what we know to destroy cancer or help to treat cancers through immunotherapy. Okay, so whenever we talk about acquired immunity, again, this is the third line of defense. This is an immunity that develops only with exposure to various antigens in one of two ways, either through infection or through medical intervention. So with that in mind, we can really talk about kind of two types of acquired immunity: active immunity and passive immunity. Let's take a look at the difference between active and passive acquired immunity. In active immunity, just like the name suggests, the individual is going to produce the antibodies against the antigen. They are actively going to produce antibodies whenever they are either infected naturally or infected by artificial means. So active immunity, again, can occur either naturally whenever we become infected, or it can be stimulated artificially by immunization. Let's take, for example, the seasonal flu. So with the seasonal flu, I could become infected by contracting the flu virus myself, and my immune system then engages, and the immune system fights off or wards off the flu virus. Or I can be receive a vaccination or an immunization that can help me to have the immune response before I get an active pathogen in my body. So with these, artificially stimulated by immunization or vaccination, what happens is the patient is either given dead pathogens that can't cause any disease or pieces of the dead pathogens. They will then have the acquired immune response occur exactly the same as what we previously discussed. Yet these pathogens are dead, or they're pieces of pathogens, so they can't cause any problem. In addition, what can be administered in the vaccination or immunization is attenuated or weak pathogens. So attenuated pathogens have usually been either chemically or heat treated to weaken them to the point where even if they were to infect your body, and they're going to be given through the vaccination, they're so weak that they can't cause any damage to cells, but your body's still going to respond to them as if they did. Good examples of active immunity acquired by immunization or vaccination is the seasonal flu vaccine. Now, in addition to active immunity, there's also that passive immunity. Passive immunity involves antibodies that are produced in a body other than your own. So you're not producing the antibodies yourself. Instead, the body of some other individual produced the antibodies, and now you're receiving the antibodies. Or I guess I should say that they're produced through genetic recombination. So, for example, in passive immunity, infants will have passive immunity because they're born with antibodies that are transferred to them through the placenta of the mother. So the mother gets infected, maybe she gets the seasonal flu or she gets the rhinovirus. The acquired immune response is set off. She produces the antibodies, IgG antibodies, clears the virus from her own system. Now she has these antibodies present in her blood. The antibodies can cross over into the blood of the newborn and even the fetus while it's developing, and they can immediately respond if the virus or bacterium, whatever the pathogen is, enters the baby's body. The baby, what's essential here is the baby didn't produce those antibodies. The antibodies were produced by mom. She transferred them to the baby. Since the baby didn't produce the antibodies, that would be a kind of passive immunity. Also, individuals can receive passive immunity by given prepared antibodies via injection. So an example of that would be, let's say, for example, an individual is bitten by, let's say, for example, a rattlesnake. So what can happen is then that venom can act as a toxin that can cause a lot of damage in the cells and even result in the death of the individual. So what do? We give the individual anti-venom. Anti-venom actually consists of antibodies that were produced either by genetic recombination or in the body of an individual that's been infected. And then through the injection, through the vaccine, the antibody is delivered into the body of the individual who's been bitten by the snake. Those antibodies will work to help to remove the toxin from the system. Okay, we're going to wrap this up very quickly by talking about some immune system disorders. The first disorder we want to talk about are allergies. Allergies are exaggerated responses to substances, allergens, that are generally not harmful. So this is things like pollen, dust. They're generally not harmful, although they can, be delivered into the system. And what ends up happening is your body, if you have this allergy, treats them as if they are actually potentially harmful. So the immune response is engaged. And it's usually exaggerated. Usually what happens initially is whenever a pathogen enters the body, what happens, there's kind of a localized response. You get that inflammation that occurs that helps deliver the neutrophils and the macrophages to the tissues to help remove the pathogen. But in this case, what happens is, and again, this is triggered by things like dust or mites or pollen. Many of you may have allergies, you know what I'm talking about there. But instead of just having that localized response, there is a systemic, a system-wide response. So it's an exaggerated response, and you get this dilation of blood vessels all over the body. So that the blood now is, instead of being directed into one place or another, blood is trying to be directed to all over. Well, what can happen is with this dilation is there can be a very significant drop in blood pressure. And with that blood pressure drop, there's not enough blood pressure. Remember whenever we talked about capillary exchange, blood pressure is very important in helping to move the fluids between the blood vessels and the tissues. So there can be a drop in blood pressure. We don't get good capillary exchange, and that can result in what we refer to in this case as anaphylactic shock. Anaphylactic shock is an immediate allergic response that occurs because an allergen has entered the blood. So if you think about it, what is shock? Shock is a rapid decrease in blood pressure that causes a drop in capillary exchange, so we're not getting good capillary exchange and the cells aren't getting what they need. It's anaphylactic because this shock is the result of the presence of an antigen. So what kinds of things can cause these bee stings? What kind of exaggerated responses? Bee stings, maybe some of you have to carry an EpiPen with you because you'll have this exaggerated response to the protein that's in the bee sting. For most people, it doesn't cause a big problem, but in some people, they have this exaggerated response to it. Others can have exaggerated responses to drugs like penicillin, and there are peanut allergies out there. People can even have these responses to peanut butter or even the smell of peanut butter. The symptoms of anaphylactic shock, obviously there's that drop in blood pressure that we see. That may result in fainting, dizziness, lightheadedness because of the lack of proper blood supply into the tissues of the brain, and that can cause the fainting and lightheadedness. Lack of appropriate blood supply can lead to anxiety and confusion and even slurred speech. This sounds a lot like stroke, doesn't it? Because again, the same thing's happening with a stroke. We're having a decrease in the blood supply to the brain, and it all depends on what part of the brain is getting the decrease in the blood supply. So we're going to see these in association with a lack of appropriate blood supply to the brain. Okay, next up we take a quick look at autoimmune disorders. These occur when the immune system mistakenly attacks and destroys healthy body tissue. There are over 80 different types of autoimmune disorders. Again, this is going to be the immune system is going to attack itself, self. So the immune system is attacking itself. There are over 80 different kinds. We can't take a look at all of them. One we can take a look at is lupus, or what clinically is called systemic lupus erythematosus, or SLE. This is a chronic inflammatory autoimmune disease or disorder that may affect many organs, including the skin, the joints, and internal organs. This happens because the B cells are producing anti-DNA antibodies. And when you think about it, let's say, for example, with the skin, because the skin is oftentimes a first sign or symptom of lupus. So what happens is we know that skin cells constantly get abraded and die, and when they do, they release DNA into the system. And these anti-DNA antibodies then will have this blood vessels are going to dilate, deliver the antibodies. There's going to be the inflammatory response. The macrophages are going to go in there. They're going to start to engulf and destroy skin cells, and as a result of that, now they're going to attack even healthy skin cells, and that's going to cause the problems that we see with the lesions on the surface of the skin with lupus. So again, this is an autoimmune disorder. Another immune system disorder is an immune deficiency disorder. This is different than autoimmune. This occurs whenever the body's immune response is either reduced or absent. So you don't have a response against self. Now you have a reduced response, immunodeficiency. Most commonly, these disorders occur whenever specialized white blood cells called the T or B lymphocytes, or both that we've already talked about, don't work as they should, or whenever your body doesn't produce enough antibodies. Well, why wouldn't that happen? Well, a good example of that is what happens in AIDS, or acquired immune deficiency syndrome. AIDS is caused by the HIV virus, the human immune deficiency virus. And this virus attacks mostly T cells. Now, it can attack other cells as well, but it attacks the T cells. And what's important about the T cells? Remember, those T helper cells are the ones that once engaged by the antigen presenting cell or the B cell, they're the ones that send out the signals to cause the rest of the response to occur. As a result, if the T cells are busy being infected by a virus, they're not going to respond correctly. They're not going to produce enough cytokines or cytokines at all. And we're not going to get the antibody response. We're not going to get the macrophage response that we saw in the previous explanations and videos. So we're going to have a kind of a dampened response, and that means it's going to be much more difficult to clear the pathogen from the system. And as I said, macrophages, B cells, and monocytes may also be infected, further dampening the immune response. Okay, that's our second screencast lecture on the immune system, and we'll see you next time.